Hello everyone, welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture we talked about different enzymes required for the digestion of proteins, carbohydrates, fats. Today we are going to consolidate all the enzymes at one slide so that you could easily pick up that which enzyme is going to act on which substrate and what are the end products. Salivary amylase, it is a part of salivary secretion, also known as ptyalin. It is secreted from mouth and buccal cavity. It acts on starch and the end product is disaccharides. I mean, it mainly convert starch into maltose. Along with some salivary amylase, there are gastric amylases in very lesser amount and pancreatic amylases. Pepsinogen is converted into its active form pepsin. The source is stomach or the gastric glands. The substrate is protein and the end product is large peptides. Pepsin, it is an endopeptidase and it basically makes converts proteins into proteoses and peptones, which are large peptides. Then pancreatic juice, it includes pancreatic amylase, source is small intestine, substrate is starch, end product is disaccharides. Trypsinogen is converted into trypsin, which is the active form, small intestine, substrate is protein, and the end product is large peptide. Chymotrypsin, active form of chymotrypsinogen, small intestine, substrate is protein, end product is large peptide. Proelastase, proelastase. In the presence of trypsin is converted into elastase. In the presence of trypsin converts into elastase. Source is small intestine. Substrate is elastin. Elastin fibers mean they are major carbohydratic fibers, protein fibers, major cross-linked protein fibers, cross-linked oligopeptides. Carboxypeptidase and aminopeptidase, they are exopeptidases, and they act on the terminals. They act on terminals. Small intestine, the substrate is large peptide, and the end product is oligopeptide. Carboxypeptidase, its substrate is large peptide, and the end product is amino acid. Aminopeptidases, its substrate is large peptide, and it converts into oligopeptides. Oligopeptide means a peptide chain having 2 to 6 amino acids or peptide linkages. Peptide linkages. Okay. Lipases secrete from small intestine. Substrate is triglycerides, fatty acids, and glycerol, and the end product is monoglyceride. Nucleases from a small intestine. Nucleic acids, substrate is nucleic acid, and the end product is nucleotides. Okay. Nucleases mainly include nucleotidase and nucleosidase. Nucleotidase and nucleosidase. Nucleotidase convert nucleotides into nucleoside plus phosphate group. It mainly converts nucleotide into nucleoside plus phosphate group. And nucleoside is, it break down the nucleoside into pentose sugar and nitrogen base. Okay. Now moving to the next slide. Succus entericus, or the intestinal juice. It includes entropeptidase or enterokinase secreted from duodenum, proximal wall of duodenum. Acts on trypsinogen and into trypsin. Acts on trypsinogen and convert into trypsin. Peptidases from duodenum. Substrate is oligopeptide, converts into amino acids. So the amino acids, they are majorly formed from peptidase, especially the carboxypeptidase. Disaccharidases, duodenum. Substrate is disaccharides and the end product is monosaccharides. Nucleotidase and nucleosidase I have already discussed. Procarboxypeptidase, it basically break the peptide bond from C terminal or carboxy terminal. It breaks the peptide bond from the C terminal or the carboxy terminal. It is a exopeptidase. Procarboxypeptidase is a exopeptidase as it cleaves the peptide bond from C terminal or carboxy terminal. Aminopeptidase, it is also a exopeptidase. It breaks the peptide bond from N terminal or amino terminal. Okay. Aminopeptidase cleaves terminal bond from N terminal, which is also a exopeptidase. Then tripeptidase and dipeptidases act on tri and dipeptides. Tri and dipeptidases acts on tri and dipeptides. I hope everything is clear. All proteases, they are basically divided into endopeptidases and exopeptidases as already discussed. In exopeptidases, you have to remember two names, carboxy and amino, while all are endopeptidases. End product of protein digestion is amino acid. End product of fat digestion is fatty acid and glycerol. Okay. Now, Ebner's gland, as we have studied about Brunner's gland, goblet cells. Ebner's gland on the tongue, which secrete lingual lipase. Ebner's gland on the tongue, which secrete lingual lipase. Okay. Lipases, they are lingual lipase, gastric lipase, intestinal lipase, pancreatic lipase. Pancreatic lipase also known as T-epsin. Known as T-epsin. Okay. Gastric lipase, they are active in children as pancreas are insufficient. Mean pancreatic lipase are insufficient in children. In children. That's why gastric lipase are more gastric lipase are more in children. Okay. Intestinal lipase, they basically have negligible role in fat digestion. Mainly pancreatic lipase, they are concerned with the fat digestion. Okay. The rest everything is already discussed with you in the previous slides. This is absorption of food we have already discussed. Maximum water absorption takes place in the upper part of the small intestine passively through passive diffusion. Okay, passive diffusion. Absorption in colon. Colon absorbs water from the undigested food. Very important. Colon mainly absorbs water from the undigested food. Due to hostra, I have already discussed hostra, villi, microvilli, brush border. Due to hostra, the water absorbing capacity of colon increases and it efficiently increases absorption of water. Colon absorbs water from undigested food due to hostra. The water absorbing surface of the colon increases and it efficiently increases absorption of water. The excreta of rabbit is given out of the body in the form of small pellets. The excreta of rabbit is given out of from the body in the form of small pellets. The process of removal of undigested food is known as defecation. The process of defecation is involuntary in rabbit. It is involuntary in rabbit, though it is voluntary in most mammals. The process of defecation is involuntary in rabbit, meaning it is not in the control of rabbit, and though it is voluntary in most of the animals. Symbiotic bacteria is found in colon. Bacteria synthesizes vitamins K, vitamin B1, vitamin B2. Undigested food goes into the rectum where it gets converted into feces that contains mainly water and solid matter. Undigested food goes into the rectum where it is converted into feces, which mainly contains water and solid matter. Solid matter contains dead bacteria 30%, fat 10 to 12%, proteins 2 to 4%, and others. Solid matter mainly contains dead bacteria 30%, fat 10 to 12%, proteins 2 to 4%, and others. These feces ejected outside through anus. All these feces, they are ejected outside through anus. Substances move along the concentration gradient without expenditure of energy, that is known as what? Passive. And the substances which move against the concentration gradient with the involvement of energy is known as what? Active transport. Okay. Now, moving to the next slide. In morning, the excreta of the rabbit is in the form of semi-solid pellets. It has more amount of undigested cellulose in it. More amount of undigested cellulose in it. Cellulose is a colloid substance. Colloids have the capacity to bind water on their surface. So complete absorption of water is not possible in the intestine. To completely digest the cellulose, rabbit again ingests the semi-solid excreta. So again digestion of cellulose takes place in the cecum. Cellulose is a colloid substance. It has capacity to bind water on their surface. So complete absorption of water is not possible in intestine. To completely digest the cellulose, rabbit again ingests the semi-solid excreta. So again digestion of cellulose takes place in cecum. The final digestion takes place in cecum. In the evening of excreta, in the evening, the excreta of rabbit in the form of solid, dry pellets. These have less amount of undigested cellulose in them. This nature of rabbit to eat its own excreta is known as caprophagy or cecotrophy, or is known as pseudorumination. Double circulation of food through the alimentary canal is termed cecotrophy. Double circulation of food through alimentary canal is termed as cecotrophy. As it is first giving out the semi-solid pellets, then again it is taking its own semi-solid pellets as a food material and then digesting it in the final digestion taking place in the cecum. Food of rabbit mainly consists of cellulose, so this activity is necessary for rabbit. Brown color of excreta is due to two pigments, stercobilin and urobilin. Both of them are formed due to the degradation of bilirubin. False smell of excreta is due to indole, very important, scatol and tryptophan, methane, ammonia and H2S. Brown color of excreta is due to two pigments, stercobilin and urobilin. Both of them are formed due to the degradation of bilirubin. False smell of excreta is due to indole, scatol, tryptophan, methane, ammonia and H2S. These are found in colon due to the decomposition of amino acids of bacteria. Decomposition of by bacteria. Pellets of rabbit don't have a foul smell because it has minimum amount of proteins in it. Carnivores have excess protein rich diet, so the excreta is highly foul smelling. Pellets of rabbit don't have a foul smell because it has a minimum amount of protein in its diet. Carnivores have excess protein rich diet, so their excreta is highly foul smelling. Okay. Compound stomach. Stomach of ruminant animals made up of four chambers. Rumen is the largest, reticulum is the smallest, omasum, abomasum. Rumen, reticulum, omasum, abomasum. Okay. Rumen is the largest, reticulum is the smallest, omasum and abomasum. Gastric juice is secreted by abomasum, which is known as what? True stomach. True stomach. Inner surface of rumen and reticulum lined by keratinized epithelium. Symbiotic bacteria found in rumen and reticulum. Voluntary muscles found in rumen and esophagus. Hence, reverse peristalsis are found in rumen and esophagus, which is controlled by willpower of animal. Reverse peristalsis are found in rumen and esophagus, which is controlled by willpower of animal. Voluntary muscles are found in rumen and esophagus. Omasum is absent in camel and deer. Omasum is absent in camel and deer. So rumen is the largest, reticulum is the smallest, omasum, abomasum is the true stomach. Gastric juice is secreted by abomasum, that is why it is known as what? True stomach. Inner surface of the rumen and reticulum lined by keratinized epithelium. Symbiotic bacteria found in rumen and reticulum. Voluntary muscles are present in rumen and esophagus. That is why the peristalsis happened is voluntary. Reverse peristalsis, voluntary. Rumen and esophagus, which is controlled by willpower of animals, of animal. Omasum is absent in camel and deer. Calorific value. The amount of heat liberated from the complete combustion of 1 gram of food in a bomb calorimeter, which is a closed metallic chamber filled with oxygen, and its gross calorific value or gross energy value. The amount of heat liberated from the complete combustion of 1 gram of food in a bomb calorimeter, a closed metal chamber filled with O2, and its sorry. The actual amount of energy liberated in the human body due to combustion of 1 gram of food is the physiologic value of the food. So the gross calorific value or the gross energy value of carbohydrates is 4.1, while the physiologic value is 4. Proteins, I mean all these food substances when they are you know when they undergo complete combustion in a bomb calorimeter, then it is the gross energy value or the calorific value. While when they are physiologically combusted inside the human body, then it is known as what physiologic value. So carbohydrate, the physiologic value is lesser than the gross calorific value. Proteins, they are also same. The physiologic value is lesser than the gross calorific value. Fats, same here. The gross calorific value for the fat is 9.45. They all are basically the amount of energy liberated in kilocalories when 1 gram of the substance is combusted. Okay, the physiologic value and the gross calorific value or the gross energy value. Okay, assimilation. The use of absorbed digested food by the body is termed as assimilation. Amino acids synthesize proteins, which in turn synthesize enzymes and new protoplasm. Glucose, fatty acids, glycerol on oxidation provide energy. So the fate of glucose, fatty acids, and glycerol is to provide energy, while amino acid fate is to synthesize proteins. These proteins synthesize enzymes and new protoplasm. So they help in repair, regeneration, recycling, cellular division, and resynthesis. Okay. What is protein energy malnutrition? Kwashiorkor and marasmus, these two diseases, they are kept under this category. Kwashiorkor, it is a protein deficiency disease. It commonly affects the infants and children between 1 to 3 years of age. Symptoms: underweight, stunted growth, poor brain development, loss of appetite, anemia, edema on lower leg and face. Symptoms are underweight, stunted growth, poor brain development, loss of appetite, anemia, edema in lower leg, edema means the swelling in lower leg and face. Swelling due to retention of water, tissue fluid. Kwashiorkor is a protein deficiency disease. It commonly affects infants and children between 1 to 3 years of age. Symptoms are underweight, stunted growth, poor brain development, loss of appetite, anemia, edema in lower leg and face. Here is proteins are necessary for growth, repair of tissue, and for body defense. Therefore, adequate amount of proteins must present in the diet. Daily requirement: 1 gram of protein per kg body weight in adult. 2 gram of protein per kg body weight in growing children. Daily requirement is 1 gram of protein per kg body weight in adult, and in growing children 2 gram protein per kg body weight in growing children. Sources: cereal pulses, meat, fish, milk, groundnut, peas, leafy vegetables, etc. Marasmus, it is a protein, energy malnutrition, or deficiency of protein and total food calorific value. Symptoms: impairs physical growth, subcutaneous fat disappears, ribs become very prominent, lips become thin, and skin becomes dry, thin and wrinkled. There is no edema on leg and face, but loss of weight occurs. Loss of weight occurs in case of marasmus. Cure: diet with adequate proteins and proper calorific value should be given to the infants. Same as kwashiorkor. I will show the pictures in the next coming session. So I will show the pictures of kwashiorkor, marasmus, and we will discuss others diseases related to the alimentary canal like malabsorption syndrome and other diseases. Till then, bye bye. Take care. Stay safe. Stay healthy, everyone. Bye.